I'm going to talk about the Amino Acid Starter Kit. And this is a kit that we use to introduce high school students to the concept of a protein. Generally, when you say the word protein to a bunch of high school kids, the first thing they will think about is hamburger or their diet. They think of protein as something that they eat along with carbohydrates and fat and things of that sort. And while that's certainly true, that's not at all what we're trying to convey to them with this kit. We want them to begin to think at the molecular level about a protein, what it is, what it's made up of, and why they assume these complicated three-dimensional shapes. So in this activity, we have what we call a mini tuber, which is just a foam-covered wire. And on that tuber, on that mini tuber, we can add what we call side chains. But a protein is made up of amino acids. And in fact, let me give you the CBM's definition of a protein. A protein is simply a linear sequence of amino acids that spontaneously fold up into a complex 3D shape following basic principles of chemistry. So that's sort of a three-phrase definition of a protein. And that's how we want students to begin to think of proteins. So this model, this amino acid starter kit, is a great way to sort of attach some meaning to that definition of a protein. So let me put this model of a protein down here for a minute. And first remind you that proteins are made of amino acids. So here's another physical model. This is what we call a molymod model of an amino acid. It's got a amino group here, this blue nitrogen. Chemists will call that an amino group. And then on the other end, you have these two oxygens and a carbon. Chemists will call this a carboxylic acid functional group. So you've got an amino acid, and these two functional groups are joined by a central black carbon atom. Call this the alpha carbon. And there are 20 of these 20 different amino acids that commonly make up proteins. And on the alpha carbon, we have represented generically the side chain, or sometimes the R group of an amino acid. And we've represented it just with this pink colored sphere. So understand that there are 20 different chemical structures that are attached to this alpha carbon. And that's what makes up the 20 amino acids. All right, hold that thought for just a second. And let me just say that a protein then is made by joining consecutive amino acids together to make a long sequence of amino acids. Each one of these side chains represents one amino acid that's been joined one to another to another. All right, so side chains. What are the side chains that get added to this alpha carbon? That's what we have here. In the amino acid starter kit, you'll find a little bag of the 20 amino acid side chains. And let me just grab one here. This looks like this is aspartic acid, and it has a red clip on it. That's to signify that this is a negatively charged amino acid, because it has a carboxylic acid functional group here. And I can tell that, I don't know if you can see this in the video, but embedded in this side chain, there are color-coded atoms that correspond to the atoms that make up that side chain. So there are 2 red oxygens and there are 2 gray carbons that make up aspartic acid. And the first thing you will do is you will dump out the little baggie of the 20 amino acids, and using a chart that comes with this kit, you will identify each one of those shapes, and you'll say, oh, that's threonine. it has a white clip, which means it's polar but uncharged. You'll find lots of amino acids with yellow clips. These are hydrophobic amino acids that go up here at the top of the chart. All right, so the amino acid starter kit then consists basically of a tuber, mini tuber like this. It's got red and blue end caps. Can anyone guess why we have red or blue and red? I should turn it around like this, blue and red. This is the N-terminal end of the protein, where you have a blue amino group or nitrogen that starts this protein off. And then the amino acids are joined together until you get to the C-terminal end, where we have a red end cap corresponding to the red oxygens of the carboxylic acid group. So you have a mini tuber backbone. And in this kit then, to do the activity that's described in this kit, you're going to start adding side chains from this pie chart onto the mini tuber to create this kind of a representation of a protein. Remember the definition that I gave you a few minutes ago? A protein is simply a linear sequence of amino acids. So that's what we have here. We have here valine, aspartic acid, leucine, arginine, etc., joined together in a very specific sequence. Second part of the definition is this sequence of amino acids will spontaneously fold up into a compact 3D structure, right? And it does that following basic principles of chemistry. All right? Now, what are those basic principles of chemistry? Because if you're going to fold your protein into a shape that makes sense, you're going to have to know what those principles of chemistry are. And that brings us to yet another model. And this is maybe the most important basic model that we use. This was one of the first models that 3DMD ever developed and marketed. It's called a water cup. And the main thing you're going to find in a water cup are water molecules. So water, we are a water-based life form. 70% of the mass of our cells is water. So if we want to understand how this protein, which is made inside of a cell, which is 70% water, if we want to understand the principles of chemistry that drive it into this shape, we have to understand the properties of water. So water, its most significant property is that it's polar or hydrophilic. Those words mean the same thing. So if we looked carefully at a water molecule made up of 3 atoms, a red oxygen and 2 white hydrogens. So all of the electrons that are contributed by these 3 atoms to this molecule, they are unevenly distributed around these 3 atoms. And that's the fundamental property of water. The electrons, which are negatively charged, spend a little more time around the oxygen than they should, and therefore the oxygen becomes just a little partially negatively charged. At the same time, the electrons are not around the hydrogen as often as they should be, and these become slightly positively charged. So due to that asymmetric distribution of electrons, when one water molecule comes close to another one, they're able to interact through this hydrogen bonding, partially negative oxygen, partially positive hydrogen. So water sticks to itself and any other hydrophilic molecule. All right? Now, we could go ahead and talk about ice, states of matter, and all sorts of stuff, but we're talking about proteins. So I just want to point out how the principles of chemistry in this water cup can be applied to the amino acid side chains of a protein. So we've established the polar nature of water. You'll also find in a water cup a very different kind of molecule. This is a hydrocarbon. And the electrons around all of these atoms that make up a hydrocarbon are evenly distributed around these 8 atoms. And therefore, there's no way for hydrophilic water to interact with hydrophobic or nonpolar hydrocarbons. So we've established the chemical principle of being either hydrophobic or hydrophilic. And we're going to add those to the side chains in just a second, but I also want to point out the water cup also has a sodium chloride ion pair. This is table salt. This is sodium chloride. Put sodium chloride in water, it dissolves, and that's because this negatively charged chloride ion is going to be hydrated by the hydrogen of a water, and at the same time the positive sodium ion will be hydrated by the oxygen of another water, and these two ions will just be hydrated or solvated and will dissolve in the water. All right, so let's now take these principles of chemistry and apply them to these side chains. So first of all, the hydrocarbon is like these yellow clips up at the top. These are amino acid side chains that are made up only of carbon. So they're like hydrocarbons. Now, there are always exceptions in biology. So one of these hydrophobic side chains, tryptophan, has a nitrogen in it. But that's the only exception. All of the rest of these yellow clips have only hydrogen. Only carbon. And I just realized that's not true, but I'm not going to take time to tell you another exception to that. All right, so the hydrocarbon is like the yellow side chains. Water is like these white clips down here that are polar but uncharged. There are 2 basic amino acids that are positively charged, and that's very much like the sodium ion of the ion pair. And over here, the negatively charged aspartic acid and glutamic acid, those carry a net negative charge, and they're like the chloride ion. This is where our color coding breaks down, because some chemists a long time ago said that chloride should be green. So you'll have to forgive us for that color inconsistency. All right, so those are the principles of chemistry then that are going to determine how these different amino acid side chains interact with each other. So now let's go back to our protein, which is a linear sequence of amino acids. It's going to fold up into a complex 3D shape following those principles of chemistry. And remember, this is being made in a cell, which is 70% water, which is polar. So one of the first principles of chemistry that are going to drive this linear unfolded protein into a compact folded protein is the tendency for these yellow clipped hydrophobic amino acids to minimize their interaction with water. So I'm just folding this tuber into a shape in which I'm collecting all of the yellow hydrophobic amino acids, side chains, into this hydrophobic cluster in the middle, and then the rest of the protein kind of wraps around that hydrophobic core to shield it from the water that's floating out here. So that's one principle of chemistry. But what appears to be a very trivial, simple explanation gets a little harder then when you realize that you have to simultaneously satisfy several principles of chemistry. So another principle is that opposite charges attract, just like sodium and chloride form an ion pair. Here we have a positively charged arginine. Here we have a negatively charged aspartic acid. So they would really like to come close to one another to form what we call a salt bridge. This is a charge-charge interaction between a negatively charged aspartic acid, positively charged arginine. But in doing that, I have exposed to this water these 2 hydrophobic amino acids. So the trick then is to constantly sort of massage your protein. You have to kind of work with your protein to find a structure that simultaneously satisfies several different principles of chemistry. So I can find a shape that at the same time keeps these two together in a so-called salt bridge, but at the same time delivers these two hydrophobic residues back into this hydrophobic cluster. All right, so there's one modification. Now, this is what makes this an interesting exercise, and also can be a frustrating exercise depending on how lucky or unlucky you are. Because we set this up so that there are two positive and two negative side chains on this protein. So here is another positively charged side chain. Here's another negatively charged one. So now I'm going to somehow try to further modify my protein. You know, it's easy to do when you're focused just on one thing. I can easily bring these two together to make a second salt bridge. Here's my new salt bridge. Here's my original salt bridge. But once again, I've exposed this hydrophobic group, but we'll just simply tuck that group back under here into the hydrophobic cluster. So now I have a structure that simultaneously has a hydrophobic cluster and two salt bridges. So then, just because we like to irritate students, we add one more feature, and that is there's an amino acid called cysteine, and it's represented with green clips. And that's because cysteine is unique in that it can form a covalent disulfide bond with another cysteine. So here's a green cysteine, here's a green cysteine. So all we have to do to complete our folding is to bring those two cysteines close enough together that you can imagine a disulfide bond. And I think I've done that without causing too much damage to my hydrophobic cluster, although you see what's happened here. All right, so I'm not exactly sure how well you're able to see all of this in this video, but I hope you can imagine that the real power of this amino acid starter kit is that it gives you a way to think about the principles of chemistry that drive these long linear sequences of amino acids into a specific three-dimensional shape. And then, of course, in proteins, there's something called the structure-function relationship. So what we've learned over the years as we've begun determining the structures of proteins, we see over and over and over again that the structure of the protein often explains to us how that protein performs its function. So not only do proteins exist as a specific sequence of amino acids that always fold into the same shape, and your ears should pick up whenever you hear anyone say always in biology, because biology is a much more interesting science than just a bunch of rules that are always followed. Biology has principles that are usually followed. So what I'm trying to say is that proteins don't always fold up into complex shapes, but usually they do. But that's a protein. It gives you a way to understand that three-phase, three-part definition of a protein as a long linear sequence of amino acids that fold up into this complex 3D shape. And they don't do it just willy-nilly. They don't do it because they want to, although in a sense they do. There's a law of thermodynamics that explains why they appear to have this tendency to collapse on themselves to spontaneously make a compact structure. But they do so because they're obeying these universal principles of chemistry. That's what we want students to think about when they hear the word protein. So since we taped this video on the amino acid starter kit and protein folding, a lot has happened. Coronavirus has happened, and all of us are experiencing a different world. Many of you are facing a world in which you're going to have to be doing a lot of remote teaching. So what 3D Molecular Designs, has done recently is they've taken some of our most popular kits, like this Amino Acid Starter Kit, and we've packaged them into what we call student modeling packs. So these are sort of minimalistic components of these various hands-on kits that we imagine your kids being able to use at home or in some remote situation where it's not just a teacher in front of a class of students. So I want to just show you the amino acid student modeling pack, the protein student modeling pack, which includes the amino acid starter kit, which we just talked about, as well as another kit called the enzymes in action. So let me start with, let me just show you what you get. In the protein pack, you get a tuber, and on the tuber you have some of these clips that normally would have the amino acid side chains attached to them. So because we want to keep the cost of these individual modeling packs down, we're not including the injection molded clips, but what we are including is this sheet of paper which shows the 20 amino acid side chains. And if you cut these out, then you end up with this, which is a bunch of side chains. And then if you just take one of those side chains and you fold the bottom a little bit, this looks like proline. And proline is hydrophobic, so it has a yellow clip. And you then just attach that into a yellow clip. And you have the same thing that you might have experienced in a classroom using the Amino Acid Starter Kit. You take this tuber and you imagine these side chains here, or you can add the side chains in advance, and you're going to fold this into a protein like this. So you would say, here are two hydrophobic residues here and here. So you'd collect all the hydrophobic residues here. This is a little shorter than the one that we used. This has about 8 to 10 amino acids. We used an activity in which we had 15 amino acids on a tuber. So this has only one salt bridge. Look here, I'm really lucky because I've got a red opposite of blue. Here's a green cysteine, here's a green cysteine. So if I pull this back here, this turned out to be very easy. So there's a protein shape that follows those principles of chemistry. All I have to do now is further elaborate. I see a yellow clip here. So here's an isoleucine. So I might fold this one, insert that right in there, and I could then construct the primary sequence of this amino acid. Ultimately, you get a folded tuber like this. So that's one half of the protein student modeling pack. The other half is something that we didn't discuss in the last video, but it's what we call the enzymes in action kit. So this is a very schematic model of a protein, actually of an enzyme to be more precise. And this is what you get. You get a gray colored enzyme, and then you get these various substrates and inhibitors. Okay? And this is what they look like when you take them out of the packet. But then when you do a little coloring or drawing on them, you end up with this. So we've now decorated our enzyme so that there's a hydrophobic pocket right here at the bottom of this enzyme active site. And there's a negatively charged side chain over here. There's a positively charged side chain over here. And then we've decorated two different substrates. The red one we'll see will fit very nicely into this enzyme substrate. So the shape is consistent with the shape of this active site, and plus the charges make sense as well. We have minus versus plus, so we have two salt bridges, and we have a hydrophobic region here, which is accommodating the hydrophobic part of the substrate. We have another substrate that looks to be the right shape, but you can see this would not be a good substrate for this enzyme because it doesn't have a hydrophobic portion here, and the charges would actually lead to charge repulsion rather than attraction. You see there's a hydrophobic portion up here, so we could turn this around and match the principles of chemistry, but now you see the shape isn't quite right. So this will emphasize to students that when a substrate binds to the active site of an enzyme, it has to match both the three-dimensional shape and the chemical nature of the groups that line that active site. So those are two substrates. And now here we have an inhibitor. We have a small molecule that can bind near the active site of this enzyme. And it's a competitive inhibitor because now it won't allow this substrate to enter. Alternatively, we can talk about a non-competitive inhibitor. And this, I think, is one of the coolest things about this kit. The non-competitive inhibitor, we've always taught students that it binds not right at the active site. It binds somewhere else on the enzyme. But the consequence of that binding is that it changes the nature of the active site so that the substrate will no longer bind. Okay? So when you bind the non-competitive inhibitor at this site, you can see how it alters in subtle ways. This is not very subtle. This is pretty dramatic in your face. But that's a non-competitive inhibitor there. The last thing you can do with this kit is you can talk about anabolic versus catabolic enzymes. So we normally think of an enzyme as something that binds a substrate, like this substrate, and it cleaves the substrate into pieces. So here we have a substrate that binds, and now it has cleaved that substrate into two pieces. That's a catabolic enzyme. It's breaking things down. Here's an anabolic. Now we pretend this is an anabolic enzyme, and we load substrate 1, and then we load substrate 2, and it catalyzes the biosynthesis of some larger substrate. All right, so you bind two substrates, they go together, you pull out just one. So this is a terrific little schematic model of an enzyme which allows you to do a number of things: catabolic versus anabolic, variety of substrates, as well as competitive and non-competitive inhibitors. So this is only one of about 5 student modeling packs that 3DMD has put together very quickly here in hopes that you might be able to use some of these as individual student modeling kits in some remote learning situation. Good luck.